Chapter 3, Segment 4. Several factors regulate neurotransmitter release. Some neurotransmitter molecules that never make it to the postsynaptic membrane may bind to receptors called autoreceptors. Axodendritic is referring to a synapse in which a presynaptic axon terminal synapses onto a dendrite of the postsynaptic neuron, either by a dendritic spine or directly onto the dendrite itself. Axosomatic, referring to a synapse in which a presynaptic axon terminal synapses onto the soma of the postsynaptic neuron. Other forms of synapses include the axo-axonic synapse, which is a synapse in which a presynaptic axon terminal synapses onto another axon's terminal. Retrograde synapse, which is a signal, usually a gas neurotransmitter, which flows from the postsynaptic neuron to the presynaptic neuron, thus counter to the usual direction of synaptic communication. The dendrodendritic synapse, in which a synaptic connection forms between the dendrites of two neurons, and a non-directed synapse, which the presynaptic and postsynaptic cells are not in close apposition. Instead, a neurotransmitter is released by axonal varicosities and diffuses away to affect wide regions of tissues. Ectopic transmission is cell-to-cell communication based on a release of neurotransmitter in regions outside the traditional synapses. Varicosity is axonal swelling from which a neurotransmitter diffuses in a non-directed synapse. Neurons and synapses combine to make circuits. So you can think of a circuit as a group of neurons and their synaptic interconnections like electrical circuits. The arrangement of the specific components accomplish a particular function. A neural chain is the simplest neural circuit in which neurons are attached linearly end to end. The knee-jerk reflex is a basic surface or circuit for the stretch reflex. The stretching of the tendon below the knee leads to an upward kick of the leg. This consists of a sensory neuron, a motor neuron, and a single synapse in which the sensory neuron communicates with the motor neuron. The three factors that account for the rapidity of knee-jerk reflex is that both the sensory motor neuron axons involved are myelinated and of large diameter, so they conduct rapidly. The sensory cells synapse directly on the motor neurons, and both the central synapse and the neuromuscular junction are fast ionotropic synapses. Convergence is the phenomenon of neural connections in which many cells send signals to a single cell. Divergence is the phenomenon of neural connections in which one cell sends signals to many other cells. Gross electrical activity of the brain is readily detected, so the electrical activity of millions of cells working together combines to produce electrical potentials large enough that we can detect them at the surface of the skull. Measuring of brain waves or spontaneous brain potentials. This can be done by an EEG or an encephalogram, which is a recording of gross electrical activity of the brain recorded from electrodes that are placed on the scalp. Seizure disorders result from electrical storms in the brain. So epilepsy is a brain disorder marked by major sudden changes in the electrophysiological state of the brain that are referred to as seizures. A seizure is an epileptic episode. It features widespread synchronization of electrical activity. It has broad sections of the brain which start firing in simultaneous waves of excitation. These are evident in the EEGs as an abnormal spike and wave pattern of brain activity. Epilepsy can be caused by many abnormalities of the brain, including trauma, injury, or metabolic problems, which could predispose brain tissue to produce synchronized epileptiform activity, which can spread easily. There are several major categories of seizure disorders. This includes generalized seizures, which are characterized by loss of consciousness and symmetrical involvement of body musculature. Tonic-clonic seizures, also known as grand mal seizures, show an abnormal EEG activity, which is evident all over the brain. The person loses consciousness and makes characteristic movements, including the tonic contraction of the muscles for 1 to 2 minutes, followed by jerky rhythmic clonic contractions and relaxations. The seizure is followed by minutes or hours of confusion and sleep. The simple partial seizure, also known as absence attacks, is a more subtle variation of generalized seizures. The spike and wave EEG activity is evident for only about 5 to 15 seconds at a time. Sometimes they can occur many times per day, and the person is unaware of their environment and later cannot recall events that occurred during the episode. They do not show unusual muscle activity except for a cessation of ongoing activity and sustained staring. A complex partial seizure does not involve the entire brain and can produce a wide variety of symptoms, which is often preceded by an unusual sensation or aura. The aura is unusual sensations or premonitions that may be preceded the beginning of a seizure. So seizures can affect non-human animals. Enkindling, or a method of experimentally inducing an epileptic seizure by repeatedly stimulating a brain region, animals can receive repeated electrical stimulation that is too weak to cause a seizure on its own. Eventually, the effects of the stimuli accumulate to cause a spontaneous seizure. Many seizure disorders can be effectively controlled with the aid of anti-epileptic drugs. The event-related potentials measure changes resulting from discrete stimuli. So the gross potential changes evoked by discrete sensory stimuli, such as light, flashes, or clicks, are called ERPs. ERPs, or event-related potentials, are also called evoked potentials. They are average EEG recordings measuring brain responses to repeated presentations of a stimulus. Sensory evoked potentials have very distinctive characteristics of wave shape and latency that reflect the type of stimulus, the state of the subject, and the site of recording. Infants with impaired hearing produce reduced auditory ERPs or none at all in response to sounds. ERPs can also be used to assess brainstem imagery or damage. And finally, optogenetics, which is the use of genetic tools to induce neurons to become sensitive to light, such that experiments can excite or inhibit a cell by exposing light. Channel rhodopsin is a protein that, in response to light of the proper wavelength, opens a channel to admit sodium ions, which results in excitation of the neuron. And halorhodopsin is a protein that opens a channel to admit chloride ions, which results in the inhibition of neurons.